Okay, this is a pre-lab lecture video for experiment 10, which we're going to do in 2 weeks. By that I mean this is a 2-week experiment. So we're going to take this molecule, which is called ethyl acetoacetate, which has an ester functional group, and it also has a ketone functional group. And we're going to reduce the ketone functional group to an alcohol using the enzymes in baker's yeast, and as a side reaction of fermentation, where the baker's yeast will react with sucrose to make ethanol. That's a fermentation reaction. There are other enzymes in the baker's yeast that will actually reduce the ketone down to the alcohol. And this is a reduction reaction. And in organic chemistry, reduction basically means that you're going to add hydrogens to the molecule. And if you notice what happens in this molecule, we add a hydrogen atom to the carbon and a hydrogen atom to the oxygen. We'll talk about reductions of ketones in second semester in Organic 2. But that's how we define reduction in organic chemistry, as either loss of oxygen or, in this case, gain of hydrogens. In general chemistry, it was, Leo the lion goes grr. That means LEO, lose electrons, oxidation, GER, gain electrons, reduction. But in organic chemistry, it's a little bit harder sometimes to see the electrons, so we just say reduction as either loss of oxygen or gain of hydrogens. This gains hydrogens. So the goal of this experiment is to reduce the ketone portion of ethyl acetoacetate to a secondary alcohol. And if you look in your textbook in, I believe, chapter 10, you'll see that there's a couple of reagents that we will learn about next semester for this reduction. One of these is sodium borohydride. And sodium borohydride has a BH4 minus ion, which can be thought of as a source of H minus. Now H minus has a lone pair on the hydrogen. We're not used to seeing H minus. We're used to seeing a hydrogen atom with one electron, or maybe an H plus as a proton, but not as an H minus. So it turns out that H minus is a very strong nucleophile, as well as a very strong base. And in this case, what happens is that the H minus will actually react with the carbonyl group, or will react with a carbonyl group in a nucleophile-electrophile reaction. So if you look at a carbon-oxygen double bond, the carbon is slightly positive and the oxygen is slightly negative based on the electronegativity of oxygen and carbon. That makes the carbon that's delta plus electrophilic. And so if we have a nucleophile, what can happen is it can take this pair of electrons from the H minus and it can donate it to that carbon to form a CH bond. But if it does that, carbon's going to get 5 bonds. So at the same time we're forming this CH bond, we have to break the CO double bond and give that pair of electrons to the oxygen, which is actually okay because oxygen is more electronegative than carbon. And what happens is that we add the first hydrogen to the carbon, and then our oxygen now has, and if I put in my lone pairs here, that oxygen would now have 3 lone pairs and have a -1 charge. Well, after the hydride has reacted with all of the carbonyl, we then add in a second step, and this is typically written as then H plus. We then, after this hydride reduction is over, we then go ahead and dump in acid, and that O minus then will get protonated by the hydrogen, so that in the end we've added 2 hydrogens to our carbonyl group, and we've reduced it from a ketone, in this case, down to an alcohol. Now if I come back up here and look at the ketone portion of ethyl acetoacetate, it turns out that when I form this molecule, when I form that group, it'll add an oxygen, or it'll add two hydrogens, one to the carbon, one to the oxygen. It turns out that looking at this molecule, I generate a chiral center in it. And so when you do the reaction with sodium borohydride, you actually would get a racemic mixture of the R and the S enantiomers, because sodium borohydride is not a chiral reagent. Well, sodium borohydride, we'd have to use ether for this. It's not a particularly environmentally friendly reagent. And sodium borohydride is a fairly weak reagent. There is also a much stronger reagent called lithium aluminum hydride, which we would not use in this case because it would reduce the ester. But lithium aluminum hydride is a little pyrophoric on occasion. So these are not the most environmentally friendly reagents to use. And not only that, but when we do form this chiral center with a non-chiral reagent, you're just going to get a racemic mixture of that alcohol. So in doing the baker's yeast reduction, this is a more green reagent, more environmentally friendly, because we're using the enzymes in simple baker's yeast to reduce the ketone. We're doing this in a non-organic solvent. We're doing this in water, and we're using sugar, which is a fairly plentiful and again environmentally friendly reagent. So what happens in the main reaction is yeast reduces or reacts with sugar, sucrose, and that gets converted into ethanol by the process of fermentation. You probably have studied that in biology. Carbon dioxide is also produced in this reaction. Now, what people found, and this was in the probably early 80s, that somebody found that when they added Ethyl-acetoacetate to that fermentation, what they found was that there was another set of enzymes in the baker's yeast, and as a side product or as a side reaction, it reduced the ketone portion to the secondary alcohol. Because the ester is too stable to react. It's very difficult to reduce esters. So just the ketone gets reduced. And it turns out that the enzymes in the yeast, like most bio-organic reagents, are chiral. They're made up of one enantiomer. And so they act as a chiral reagent in this reaction. And so what you end up with is a majority of one of the enantiomers of the product alcohol that's formed. Now, it's not 100% of one of the two enantiomers. When I've analyzed this product in the past using a couple of more advanced techniques, I get about 85% of the S plus enantiomer and about 15% of the R minus, and that's typically what the literature reports for just using simple yeast that you can buy at the grocery store. So what we're going to do is we're going to be creating then an optically active compound, one that will rotate plane polarized light. And if the majority of the product is the plus, we should see a positive rotation of plane polarized light. That reaction will then not only be environmentally friendly, but it'll give us a chiral molecule. And this fits in with what we have learned, or what you are learning with chiral molecules in lecture. So we're going to use polarimetry to analyze this product. So yeast reductions really require several days and weeks, and some of these can be quite elaborate. You have to keep the yeast fed with sugar, and you have to keep them at a very specific temperature. We're not going to do that. We're just going to basically let the fermentation reaction occur, and in the past this has been fairly high yield. We don't see much unreacted ethyl-acetoacetate at all. But it's going to require that we start the reaction in week 9, which is the one that is upcoming as I'm recording this. And then we do the purification and do what is called working up the reaction. That means to do all the extractions, the purification, the analysis in week 10. So this is actually the week 10 experiment that we're just going to start in week 9, while you either have or are going to wait for your reaction to reflux for 45 minutes. So here's what you're going to do to start the fermentation process. We have these things called fermentation flasks. You're going to grab one of those, and then we have a special stopper that has a piece of rubber latex tubing at the top that we use to allow the reaction to basically bleed out the CO2 gas, and I'll explain what it does in a moment. Then you're going to weigh 30 grams of sucrose and 3.5 grams of yeast. And we're just using normal baker's yeast here, the same that you would use if you were baking bread or making rolls or whatever you were going to do with it in the kitchen. So in the fermentation reaction, carbon dioxide is produced. That's what causes the bread to rise when you're doing baking. So in this reaction, we do need to let the gas release from the flask or else it'll boil over. But we also have to keep air out of the reaction, or ethanol will turn into acetic acid, and what that will do is that'll lower the pH of the reaction. And one of the big differences between the yeast that we use to bake bread versus the yeast that we use to brew adult beverages is that the champagne yeast that you use to either make wine or brew beer can tolerate very low pHs. Baker's yeast cannot. So if we allowed oxygen to get into the reaction, we'd be producing acetic acid. The pH would lower and it would kill the yeast before we got 100%. So therefore, we need to allow the gas to release, but not allow oxygen to come back in. And so we use a fairly simple apparatus, and this has been around for probably 30 years, long before I got to John Carroll. This is Dr. Weaver's invention, but it's the best thing I've ever seen for this reaction. What we do is you simply take a piece of tubing, you put a glass ball at this end to basically seal up the tube. We put that tubing into a stopper, and then we make a small slit, which you can kind of see here, in the tubing. And so when the tubing normally sits, oxygen's not going to get into it, but when the pressure builds, that slit will allow CO2 to escape from the fermentation flask. And so this works very well. There are a couple of critical things, though, before you start your fermentation that you need to look at. You need to make sure that this slit is not cemented shut, which sometimes happens when these have been used in previous years. If the solution kind of gets up in there, then that sugar will seal this shut. So make sure this is open, and make sure that your tubing isn't clogged at any point except where the glass ball is in the top part of the tubing. So you want to make sure it's clean and that the gas will actually vent. Otherwise, this stopper will fly across the room at some point during the week, which I'd like to avoid. So start the fermentation. You're going to take 150 milliliters of warm tap water. It actually doesn't matter. You can use just hot tap water. These yeast are relatively unkillable. Usually if we take boiling water to the yeast, that'll kill them, but hot tap water won't. And you're going to put 150 milliliters of warm tap water in your flask, add the 30 grams of sugar, and then you're going to swirl that solution until all the sugar's dissolved. Okay, and then we're going to do this like what we would in the kitchen, which we're going to proof the yeast for about 30 minutes. Then you're going to add the 3.5 grams of yeast to the flask, but you're not going to swirl it. You're just going to allow the flask to sit. So you're going to put the flask, you're going to put the stopper and tubing on top of the flask. The yeast will float on top, and then after about 30 minutes, the yeast will begin the fermentation process, and this is in baking what's called proofing. And at the end of the 30 minutes, you'll see the foam start, and that tells us that the yeast is indeed active. Then after that 30-minute period, 30 to 45-minute period has passed and your yeast is now activated, what you're then going to do is quickly remove the stopper and the tubing, add 4 grams of ethyl acetoacetate to the flask. It is a liquid, so you're going to weigh it out in a beaker, and then you're going to swirl the flask and replace the stopper and the tubing. Write your name and section on the fermentation flask, and then you're going to we're going to set it aside, and we'll have about 100 of these flasks just lying around the lab. They won't be just lying around the lab. They'll be in designated places where the fermentation will occur over the course of the next week. Okay, so we're going to do this a little bit differently than the procedure says, where we mix everything together. We're going to proof it and then make sure that the yeast is activated. And this should hopefully calm down the reaction and keep it from shooting the stoppers all over the room, which sometimes happens. So the fermentation reaction will take several days, so by the next week it'll be completed. The gas evolution will have ceased, and basically the foaminess will be gone. The yeast cells should have fallen to the bottom of the flask. So when you look at your flask again, you'll see that all the yeast stuff is at the bottom of the flask, and the top is relatively clear. It might have a slight yellow color to it, but it won't be cloudy and foamy like it was the previous week. And it's important when you pick up your flask and take it back to your hood that you don't jiggle it so much that you resuspend the yeast, because it's going to be a lot easier and a lot faster to filter if the yeast stay at the bottom of the fermentation flask. If we try to filter this solution through normal filter paper, the yeast cells would run right through. They're very fine. And so what we're going to do is we're going to use a compound called Sealite, which is also called Filter Aid. And we're going to try and trap the yeast cells and get a clear filtrate. Now, Sealite turns out to be diatomaceous earth. And it's exactly as it sounds. It's made up of little diatoms, so if you study those in biology or research, then that's exactly what this is made up of. So what we do is we have to prepare a pad of Sealite in a Buchner funnel in order to filter out all the yeast cells. So to prepare that Sealite pad, what we first do is set up a normal filtration. We put a piece of filter paper in the Buchner funnel and then wet it down with some water, and that'll provide a seal. Then you're going to weigh out 8 grams of Sealite into a medium beaker, and you're going to add 20 mils of water. And 20 mils of water kind of makes this a little bit muddy, so you might have to add an additional 5 mils, and you want it to actually become a fairly thin slurry. Okay, this is kind of what you did with the column chromatography, except this is going to be a lot thinner and doesn't settle out as easily. So then to prepare the pad, we turn the vacuum off and we disconnect the vacuum tube from the filter flask. Once we've done that, we pour the Sealite and water slurry into the Buchner funnel, as I'm showing here, and you may have to add more water to the flask in small portions and swirl, and then pour that into the Buchner funnel in order to transfer as much of the Sealite as you can from the beaker. Don't overflow the Buchner funnel at this point, and don't vacuum filter yet. Okay. So now what we've done is now we've created sort of a slurry here, except that this will not do what the column did, which the solution kind of runs through it until the solution gets just above, the layer of the silica gel in the column. That won't happen here. So what we need to do is to create a uniform pad with no channels. The channels, that's where the solution will go, and so the yeast will not be filtered properly. So to create this uniform Sealite pad, what we're going to do is you're going to turn on the vacuum but leave the tubing disconnected from the filter flask like I have here. And then what you're going to do is then you're going to carefully touch the end of the tubing to the nipple on the filter flask, and as you touch that, the solution's going to then draw down. And so you want to kind of touch it and release, touch it and release, touch it and release until you've removed most of the water and left a very thin layer of water over your Sealite pad. Okay, and the most effective way to do this is simply to have the vacuum on the tubing and then kind of just touch it and remove it and touch it and remove it. Okay, so once you have this pad with a little bit of water on top, you're going to turn the vacuum off and then connect the tubing to the filter flask. Well, actually, you're not going to do that right now. But what we want to do is that we're going to filter the solution through the funnel, through the C-Light pad, and we're now ready to do that. And the reason that you want to keep the yeast as a thin layer on the bottom of the fermentation flask is that if you have a suspension of yeast, it's going to filter very slowly and it'll probably clog, and then you'll have to prepare 2 or 3 of these C-Light pads, and you only want to create one. So it's important you keep the solution as undisturbed as possible. Now, it says up here to turn off and connect. Don't connect the tubing to the filter flask yet. Empty the filter flask empty it of the C-Light solution that you just filtered. Rinse it out so it's nice and clean. Then reassemble the filter flask with the vacuum turned off, and then go ahead and connect the vacuum tubing. With the vacuum off at this point. Okay, so if you connect it up here, then you have to disconnect it to throw the solution out and rinse it out, but then connect it back up, but keep the vacuum off. So then what we want to do is carefully pour some of the liquid from the fermentation flask into the funnel, and you're trying not to disturb the C-Light pad as much as possible. And you want to add enough liquid so that the funnel's nearly full. Then you can go ahead and turn the vacuum on full blast, and the solution should start to filter. Okay, we don't want to turn the vacuum on because we didn't want to create a dry pad that had channels in it. So now that you've got solution in there and you're starting to filter, you can go ahead and turn the vacuum on, and then continue to add the solution from the fermentation flask until there's only the sludge left. So try and decant as much of the liquid off the sludge as possible. And that sludge is a yeast cell slurry. So at some point you're done, you have to put the yeast cell slurry in. But at that point, hopefully you've filtered out 80% of your solution. Once you put that yeast and cell slurry into the filter, it's probably going to let it go for about 10 minutes. And at that point, the funnel will probably clog, but it doesn't matter. At that point, you've already filtered all your solution. The solution that's been filtered will have a slight yellow color. It will hopefully be clear because we filtered out all of the yeast cells. It'll probably smell ethanolish, which means it'll smell like a combination of beer and bread. It'll have a yeasty ethanol smell. Now, we're going to extract or try to extract the acetoacetate product, the reduction product, from the water. And this hydroxy ester is what it's called, is extremely water soluble. So the first thing that we're going to do to reduce the solubility of the product in the water layer is to do a technique called salting out. This is described in the layman text. You add 30 grams of sodium chloride to the solution in the filter flask, and you're going to go ahead and swirl that until all the salt's dissolved. Now what that'll do is that'll reduce the solubility of the product in the aqueous layer so that hopefully it'll get extracted by one of the 3 ether layers that we're going to go ahead and extract with. So that's how we typically reduce an organic product's solubility, is by salting it out or by adding salt to it. Okay, and it's critical that all the salt dissolve before you put this in your separatory funnel, because if it isn't, you'll put it in your separatory funnel and you'll have all the salt and your separatory funnel will clog. So make sure you dissolve all the sodium chloride first. Then you're going to go ahead and extract that solution with 3 portions of diethyl ether, 3 20 milliliter portions of diethyl ether. And what that means is you're going to add 20 mils of ether, stopper, shake, extract, and then it'll be easy to see the 2 layers. In this case, I believe the ether layer will be the top layer. It'll certainly be fairly small compared to the aqueous layer that you used, because that's like 100 and some milliliters. So then what you're going to do is drain off the lower layer into a beaker, an Erlenmeyer flask, and then drain the 20 milliliters of the ether, the top layer, into an Erlenmeyer flask. And then you're going to place the aqueous layer back into the separatory funnel and extract again with a fresh 20 milliliters of ether. And then you're going to separate those layers, and you're going to place that new 20 milliliters of ether that you just extracted the product into, hopefully. You're going to add that to the original 20 mil Erlenmeyer flask. So then you're going to repeat this a third time. So at this point, you should have extracted the solution with 3 20 milliliter portions of ether. And so you'll have 60 milliliters of ether that hopefully contain your extracted product in an Erlenmeyer flask. Now, this is where the directions that I remember having to use in the spring vary a little bit from what's in the handout. And you can go ahead and outline what's in the handout, but we're probably going to end up using something like this because this never quite works exactly. First of all, it's extremely difficult to cut the layers because there'll be an emulsion that forms. So you want to look at the ether solution carefully. And if you see a bottom layer, it's aqueous. And we need to stick a pipette in and remove as much of that as we can. And once you've done that and there's no obvious second bottom layer, then you're going to transfer the solution to a 100 milliliter round bottom flask, and you're going to remove the ether by rotary evaporation. Now hopefully that will just leave your ethyl-acetoacetate reduction product in the round bottom. Now you might have said, hey, don't we dry before we rotary evaporate? Yes, we normally do, but this is a case where we're not going to, because drying this product is incredibly difficult. And so what we did was we tried to remove as much of the aqueous layer as we can, and then we go ahead and rotary evaporate. So you're going to transfer your solution from the round bottom flask into a small beaker. And if you have more than 4 mils of solution, then most likely your solution is fairly wet. The way to dry this is then to add about a half a scupula of sodium sulfate. Now, if you have less than 4 mils, you might add just a quarter of a scupula. And what I mean is about half as much as you would get if you just stuck the end of the scupula in and added. But what will happen is if you add sodium sulfate to this solution at this point, the sodium sulfate begins to actually draw the water out of the hydroxy ester product. And if your product is really, really wet, you'll actually see 2 layers start to form in the beaker. And those 2 layers, the top layer is your hydroxy ester product, and the bottom layer is the water that the sodium sulfate is actually now drawing out of the product. So if you see that those 2 layers start to form, then you can add another, let's say, quarter of a scupula, and if you see even more of the bottom layer form, then you're going to add probably a third quarter of a scupula. The idea here is that we want to remove and draw out as much of that liquid as we can from the product. So you can basically continue to add the sodium sulfate a little bit at a time until your top layer becomes as small as it possibly will be, and you don't see any additional formation of the bottom layer. And at this point, hopefully you've dried your product as much as possible at this step. Okay, so you're adding sodium sulfate, you're seeing a second layer, and then once you see that second layer, then add a little bit more sodium sulfate until that layer is as large as it can be, and the product layer on top is as small as it can get, because there's water in there. So at this point, remove the aqueous layer, the lower aqueous layer, via pipette. And then we're going to test the product for water using infrared spectroscopy. So since this week Dr. Simmons is in Hong Kong, I'm going to be in charge of all the labs. And so I will be kind of screening these products for the water. So what we're going to do is we're going to take an IR. Now the IR, we're looking at two regions. The first region that we'll look at is the OH band, which is about 3,400 or so wave numbers, CM-1, which is the unit for the infrared. And then we'll probably see some peaks around, let's say, 3,000 wave numbers. And then we'll see this pretty dominant peak at about 1,600. And that's peak at 1,600 wave numbers or reciprocal centimeters. That's the peak that's due to the carbonyl. And what I've found is I like the OH peak to be less intense than the carbonyl. If the OH peak is bigger than the carbonyl, then we add more sodium sulfate until we get that as much of that aqueous solution as we get as much of that water drawn out of the product, so that we then have a smaller OH and a bigger carbonyl. So that's what I'm going to be looking for. If your OH is less than your carbonyl, I'll say okay. If it's a little bit bigger, then we'll add some more sodium sulfate to it. So that's what I mean by deemed dry. It's not completely dry, but it's going to be as dry as we can get it. So once the OH is smaller than the carbonyl, then you're going to go on to the next step. If it's not, you're going to add more sodium sulfate, and I'll probably reanalyze and see what we find. So if the solution is deemed dry, then you're going to do what the procedure says and prepare a small chromatography column using cotton, a glass pipette, and adding enough alumina to that to make a column that's about 1 inch high. Then you're going to take and you're going to pass your entire solution, your entire dry solution at this point, through the alumina column, and that will hopefully remove any water that's left over. So you may have to put the solution into that pipette and use the amber bulb to push that solution through the alumina. So once that final drying occurs, you're going to collect the solution then in a tiered sample vial. That'll give you the mass of your sample. And then if your original sample wasn't necessarily dry, then you might want to get another one recorded at this point. So once we've then dried that solution, we're ready to take an optical rotation measurement to show that we do have a majority of one of the enantiomers, namely the S+, in our solution. So you're going to prepare the solution, that solution, by transferring the liquid, transfer as much as you can, to a 10 milliliter graduated cylinder. And you're then going to add enough diethyl ether to give you a volume of 10 milliliters, or whatever volume over 10 milliliters, but you need to have a minimum of 10 milliliters of solution. And then you're going to mix that solution several times by simply taking a pipette, sticking it in your 10 milliliter volumetric flask or graduate cylinder, 10 milliliter graduate cylinder. You're then going to pull some of the solution up and then kind of squirt it down and pull it up and squirt it down. That will definitely mix the solution. So then you're going to record the total volume of the solution, because we need to know how many grams of product were dissolved in how many milliliters of solution. That's the concentration that we're going to use for our optical rotation. Then you're going to take that to the instrument room. We will pour that into a cell. We'll record the optical rotation. You want to make sure you write down the number of degrees and make sure that you write down the sign. Ideally, this is the S+, so you should get a positive sign for your experimental or observed rotation. Then after that, you'll transfer the solution into a sample vial, and then if time permits, you'll rotary evaporate the solution and we'll take a GC. We'll see how that goes. The hydroxyester product that you get, I actually collect and then I'll purify it because we have we have a use for it in one of our research projects. All the other aqueous solutions can be disposed of down the drain with copious amounts of water. Now, in terms of the optical rotation measurement, you need to convert your experimental rotation into what's called a specific rotation. And so the specific rotation is called alpha, and then D, and then 20. This is what we call the specific rotation, and this is described in your lecture textbook. The D stands for the D line of sodium, which is a very specific wavelength of light that we use. It's the wavelength that comes from a sodium vapor lamp. Sodium vapor lamps used to be used in light in street lights. It has a kind of an amber glow to it. And the T is 20. The optical rotation doesn't change very much, if at all, as you change temperature, so it doesn't matter what measurement we make this at. Now, the specific rotation is a fundamental physical property for a chiral molecule. Everybody should get the same specific rotation if they have the same purity of sample. But we calculate that by taking our observed sort of experimental rotation, which we call alpha. And this is actually what you're going to get from the instrument. And we divide through by this L, which is the cell length. Now it turns out that the cell length is measured in decimeters, and the cell that we're using is 1.1 decimeters long. So that will be the cell length, and then multiplied by the concentration of the solution that you used in grams per milliliter. And that's why you needed to know what the mass of your product was, and then when you dissolve it into the 10 milliliters or into ether to make 10 milliliters total of solution, you need to know what that final volume is, because this is going to give you your concentration that you're going to multiply by the cell length, divide that product from the observed experimental rotation, and that'll give you the specific rotation of the final product. And ideally, if this was 100% of one of the products, then you would actually get the specific rotation of the S plus product, which you're going to look up in the Aldrich website, what the specific rotation should be. Now, since previous analysis says this is about an 85 to 15% R to S enantiomeric ratio, the percent enantiomeric excess, which is simply the percent of the major isomer minus the percent of the minor isomer, which in this case would be 85 minus 15. The percent enantiomeric excess that we predict in this reaction is about 60%, which means that if you want an idea of how pure your sample is, your specific rotation should be approximately 60% of the specific rotation for the pure S plus product. So whatever we determine that specific rotation to be from the literature, if your value is about 60% of that, that means your sample is very pure. If you only get about 30% of that value, then your sample is about half pure. So we can make an estimation of the purity of the product simply by analyzing the specific rotation. Okay, and that's how we'll do it for your report. Okay, so this is a 2-week experiment. First week, just set up the yeast reduction. Second week, analyze the products. If we have an overabundance of time, which I'm thinking we won't, then we may analyze the product by GC, but I think we're probably just going to analyze its purity by the specific rotation as well as the IR.